Plants turn sunlight into sugar and provide chemical energy for most living organisms on the planet, including various microbes. Through millions of years of evolution, all plants have evolved immune systems to protect themselves against the vast majority of microbial invaders, including viruses, bacteria, fungi, and fungus-like oomycetes. However, aggressive pathogens can cause serious diseases on their host plants. One of the most common and important plant diseases is powdery mildew. This disease can be found in over 10,000 plant species, including many agriculturally important crops such as wheat, barley, tomato, cucumber, grape, strawberry, and rose. Powdery mildew is caused by ascomycete fungi that belong to the order Erysipeles. These fungi are obligate biotrophic pathogens that require living hosts to complete their life cycle. Therefore, they steal nutrients from host plants and keep them alive. After landing on the surface of a host plant, a fungal spore germinates in a few hours, and then develops an opressorium, by which the fungus penetrates the plant cell wall and further differentiates a feeding structure called the historium. Establishing a functional historium in the host cell is essential for the fungus to establish a mycelial network, capable of producing asexual new spores from conidiophores in about 4 to 5 days. In about a week, massive fungal mycelial and conidiophores form an extensive network in the surface of the host tissue. In 10 to 12 days, a layer of whitish powder-like mildew gets accumulated in susceptible host plants. That's why this disease is called powdery mildew. It is important to point out that the historium is the only fungal structure in the hostile host cells, and essential for the success of the fungus. Conceivably, the historium is the Achilles heel of the fungus, because it is the only reachable target of the host defense. Based on earlier microscopic studies, the historium is separated from the host cell cytoplasm by an interficial membrane named the extrahistorial membrane, EHM for short. The EHM represents the host pathogen interface and the critical battleground for both the host and the fungus. However, the origin and biogenesis of the EHM and the molecular warfare between the host and pathogen in this battleground is largely unknown. The Xiao Laboratory at University of Maryland characterized a broad-spectrum powdery mildew resistance gene called RPW8 from the model flowering plant Arabidopsis. Plants containing the RPW8 gene locus are resistant to all infectious powdery mildew, whereas plants lacking RPW8 are susceptible. Interestingly, although novel in deduced protein structure, RPW8 utilizes a conserved immune signaling pathway to activate resistance. A key question about RPW8 is how it activates broad-spectrum resistance via the same pathway of race-specific and basal resistance. Strikingly, by analyzing the subcellular localization of a biologically functional RPW8-YFP fusion protein, it was found that the RPW8 protein is only made in the host cells invaded by powdery mildew, and specifically targeted to the surface of the fungal feeding structure, the historium. A single optical section of a confocal image of an isolated historium from host cells expressing RPW8-YFP indicates that this protein is precisely localized at the extrahistorial membrane, i.e. the EHM. RPW8 is the first protein ever identified in this mysterious yet important interficial membrane. As demonstrated by this short movie, RPW8-YFP is targeted to the EHM by membrane vesicles. Small green dots carrying RPW8-YFP are moving, decking, and then fusing to the EHM. This is a reconstituted 3D confocal image showing that the fungal opressorium penetrated the host cell wall and formed a historium inside the host cell. The historium is encased by the EHM, where RPW8 is specifically localized. Further studies show that RPW8 somehow enhances the formation of a callose-enriched cell wall-like structure encasing the historium and triggers production and accumulation of hydrogen peroxide, H2O2, in the interface. These defenses are highly historium-targeted. Thus, in terms of defense action, RPW8 functions like a molecular missile. This finding also explains well why RPW8 confers broad-spectrum resistance to powdery mildew. Simply because the RPW8 missile targets the feeding structure of all powdery mildew pathogens. Conceivably, the RPW8 protein must contain a guidance signal for EHM-specific localization. Indeed, mutational analyses identified 2 regions with the same basic residue-enriched motif, arginine or lysine, any amino acid, and arginine or lysine, to be essential for EHM targeting. Thus, this short motif may represent the zip code to the EHM. Disruption of these 2 targeted signals can abolish EHM targeting of RPW8 and its mediated resistance to powdery mildew. The identification of RPW8 as a molecular missile for antifungal immunity in plants opens up several exciting new questions for future research. How does RPW8 activate defense at the interface? What's the trafficking cue from the EHM that directs RPW8 vesicles to the EHM? How does the EHM originate? Is it from invagination of the host plasma membrane or de novo synthesized? Do host cells engage an EHM-specific membrane/protein trafficking pathway for defense? If so, what are the trafficking components involved? Finally, can one use RPW8 as a delivery vehicle to target any antimicrobial protein to the host pathogen interface, thereby engineering novel, Astorium-targeted resistance in crop plants? Research in the Xiao Lab has been supported by the National Science Foundation, USDA, NIFA, and the Gates Foundation. This video was narrated by Edward Hermy. Thanks for watching.